There we go.
There we
Recording in
go.
progress. Great. Well, thanks so much, Ronald, and thanks everyone for being here. I really appreciate the invite, and I'm delighted to be speaking to you today. Ronald, I really like that analogy of the wearing genomics on my tool belt. That's fun. I'm gonna have to use that. So yeah, I'll talk to you today about a study that we published, I guess a couple of months ago now, although it's in its proper issue now, just as of a couple of days ago. But this is a study focused on sort of our recent ability, the recent feasibility of doing sort of grand scale population genomics on rattlesnakes, as well as a deep dive into population genetic signatures in their venom gene regions, revealing a really cool and profound role of balancing selection as well as meiotic recombination in shaping venom adaptation. And so I'll start by acknowledging and thanking all of my co-authors on this study. Each person really contributed in major ways to this, this study and to the paper. This is work that I started during my time in Todd Casto's lab, and is work that is funded by National Science Foundation, as well as a Chiricahua Desert Museum grant. And so our interest in this study really began with a question that seems like it could be straightforward, but it really is perhaps more complex than we often think about, and that's how does natural selection shape venom evolution in populations being the operative word here. And the answers to this question are potentially complex because we first need to consider the origins of venom, which we understand that in snakes and pretty much any venomous species that possesses this trait, we understand that the genes that underlie the components that make up venom evolved via duplication events from a physiological gene with generalized function. We call these housekeeping genes. Commonly, this gene duplicate was a liberated target of selection that underwent very rapid evolution due to positive selection, resulting in amino acid changes to its code, and eventual neofunctionalization. It now has a function that's totally separate from its sister copy. And this process, we understand from all sorts of different organisms that this process didn't stop there, that there have been repeated bouts of duplication, in some cases gene loss, sort of carving venom gene families from sort of the marble stone of ancestral housekeeping gene starting points. And we understand that this process, there's overwhelming evidence for this process at more ancient timescales involved really strong bouts of directional positive selection. We can think of this as A to B evolution, where an ancestral population went from a state of lacking a gene duplicate to possessing this gene duplicate that eventually sort of became the proto-venom. But then once venom gene families were established, or sort of on the course of their evolution, we have populations moving from a state of less fit venom to more fit venom. And this idea and the overwhelming evidence at sort of deep timescales for this phenomenon has led to the prevailing hypothesis that venom evolves via recurrent episodes of positive selection throughout its history, both at deep and shallow timescales. Now, one issue with this hypothesis that we need to contend with when we think about things at a more contemporary timescale is that recurrent directional selection necessarily purges genetic diversity as populations fix on a particular fitness optimum or what have you. And so one issue is that this can lead to evolutionary dead ends. For example, if the fitness landscape that is basically imposing selection pressure on their venom, if that shifts away from this optimum that they've reached, that could lead to dead ends. And we understand that at contemporary, more contemporary or shallow timescales, we know that venom is really tightly incorporated into predator-prey coevolutionary antagonism, where of course the rattlesnakes that we're studying have this potent venom to help them in killing their prey. But prey species have evolved mechanisms of resistance, and these are potentially numerous. And if we consider that the rattlesnakes that we're studying are fairly generalist in their diets, they're taking a variety of different mammal species, but they'll also feed on lizards, they'll feed on birds if they can get them. some invertebrates, and so on. And so if we consider the fact that diverse prey may spell more diverse resistance mechanisms, and so it's really kind of reasonable to expect that a large degree of reciprocal selection pressure is being imposed back onto the snake predators, manifesting in their venom phenotypes. And this scenario really mirrors a lot of really famous examples of antagonistic coevolution, things like host pathogen dynamics, plant incompatibility, excuse me. The list goes on. And these are situations that often invoke balancing selection as a mechanism to actually maintain adaptive genetic diversity. We can think of balancing selection as, rather than being A to B evolution, being A plus B evolution, a mechanism that can maintain polymorphism within populations over a time. And so our question within a question here is whether genetic patterns in rattlesnake populations support a dominant role of directional positive selection. And here, populations is the operative word, because up until a handful of years ago, the sorts of analyses that we're going to dive into today simply were not possible. Rattlesnakes were kind of a challenging system, as I'm sure Mark will also touch on too. Like, rattlesnakes were pretty hard to study with any robust genomic and population genomic methods, but that narrative has changed given sort of advances in sequencing technologies and feasibility. So thankfully, that's the case. So in our study, we focused on two species within the Western rattlesnake species complex. These are the prairie rattlesnake over here in the tan distribution map, and the northern Pacific rattlesnake over here. We sampled populations from the more northern and southern, I guess more central in the case of the prairie rattlesnake here, extensive their range to enable some within species population genetic comparisons. And we sequenced the genomes of all of these individuals at fairly high coverage, calling genome-wide SNP variants, single nucleotide polymorphisms. For any of the folks here that aren't genome jockeys, we're just talking about genotypes distributed across the genome, where we're sampling at individual base pair positions. We call these using fairly standard practices, GATK, filtering using BCF tools, and a variety of other things. And we interpreted all of this variation in the context of a chromosome level genome assembly for the prairie rattlesnake that we had published from a couple of years ago. And so the beauty of this reference and its contiguity is that it enabled us to actually locate the physical locations of venom gene tandem arrays in the genome. And so all of these blue stars that you see out here on this ideogram here of the genome give you some sense of the approximate locations of the venom genes. So despite using sort of best practices, workflows for doing variant calling procedures and so on, we still wanted a way to deal with a potential problem. And that's that despite being among the most interesting regions in the genome, venom is kind of hard to study from an allelic diversity perspective. And that's because we know that they are arrayed in these complex tandem structures in narrow regions of the genome. And variation in copy number, both within and between populations and species, can lead to inference of spurious variation. That's where, say, two different gene copies end up mapping to the same position. And so variation gets counted as allelic variation, where it's actually variation at two totally distinct genetic loci. And so we needed to develop a workflow to deal with this. We didn't want to simply trust that out-of-the-box procedures for removing such bioinformatic artifacts were going to do the job, and we wanted to analyze bona fide allelic variation at orthologous sites. And so we developed a workflow to do this that kind of comes in a couple of complementary pieces. The first is to we took our input variant call file. This is just the file that contains all of our filtered genotypes across the genome. And we performed a one-tailed Hardy-Weinberg equilibrium test. And this was explicitly designed to remove variation where we found excess heterozygous sites among our entire sample. These are cases where say 70 to 80 to 90% of individuals are in our entire data set were coming up as being heterozygotes. There's no amount of selection in the world that could produce that sort of signature. Heterozygote advantages simply can't be that strong. And so we just removed those sites wholesale from the entire data set so that we were just conservatively getting rid of them. We didn't want to interpret those sorts of variants. Now, second, we performed a copy number variant detection procedure using pairwise comparisons of all of our samples against their common reference. And the idea here was to call significant copy number variants on an individual basis, quantify the proportion of individuals per population that possess that copy number variant. This could be an indel. It is an indel. It's either an insertion or deletion. We don't know which. The point is that it's present. And then we performed masking of these copy number variants on an individual basis. The idea here being that we can allow for variable presence of these copy number variants, not totally remove all variation from all individuals, but just mask the variation in individuals where this copy number variation is present. And when we do this, apologies that this plot is gnarly looking, but I'll just focus you here to this part of the plot where we're looking at a specific venom gene region where you can see that the lines, they're showing the proportion of individuals in a given population that have the presence of a CNV. We can see this is quite variable, and in some populations is to a greater extent than others. The point being that in cases like here, all of those individuals are masked for that interval, showing a really high degree of copy number variation. Okay, so with our ability to use that workflow to get rid of spurious variation and so forth, we were left with a data set across the genome that consisted of 28 million SNP variants. And we also went into our study with a handful of population genetic predictions, namely that directional positive selection, the sort of prevailing hypothesis for how venom has evolved, and again, this sort of A to B type mechanism, a classic signature of this is that as a selected allele increases in frequency, so here we have this beneficial variant, after selection, it's moved to fixation. And you can see immediately around it, the only haplotype that's present is the red haplotype. And so the only genetic variants linked to that beneficial allele, the only variation is going to be whatever came along with it on that red haplotype. And so under this sort of scenario, neutral polymorphism surrounding the selected locus is going to decrease substantially. Now, interestingly, balancing selection leaves essentially the opposite footprint, where after selection, we have a maintenance of 2, or this could even be more, different haplotypes in our population. And so linked polymorphism surrounding that balanced locus is actually going to increase. And when this process, when balancing selection persists for a long time, we call this long-term balancing selection, it can actually transcend a speciation event, where the presence of a balanced polymorphism in our 2 descendant species implies the presence of this in their common ancestor. The alternative is that there's been recurrent mutations in both descendant species after their divergence. But this is usually interpreted in the context of other evidence that is pointing towards some sort of long-term balancing event. And right, so the signature of this is the presence of these trans-species polymorphisms. So for our study, we focused primarily on 3 of the major venom gene families in the genome. These are the phospholipases A2, the snake venom serine proteinases, and the snake venom metalloproteinases. Our rationale for this is because these 3 venom families are the dominant components of the venom phenotype in vipers generally. And they also are ones that are forming tandem arrays of different genes of different number. And what's really cool, I don't know how the rattlesnakes and vipers generally figured this out, but they have, each of these venom gene families is linked to a separate microchromosome. So if we think all the way back to Gregor Mendel, this means that these venom gene families are sorting independently of one another in populations. And so selection on any one venom gene family is not necessarily inhibited by selection at another. So that's kind of a matter for another day, but it's just sort of a cool feature of their genetic architecture. And I'll mention here that we did analyze all of the venom genes in the genome that we had annotated, but we certainly focused on these major venom gene families because they're probably receiving the brunt of selective pressure. And for today, just for the sake of time, I'll talk about a case study in the snake venom metalloproteinase region, because the results in this region are really kind of illustrative generally of what we were finding in these other tandem arrays as well. So I'm going to step you through a series of genome scans where we're scooting along chromosome 9, the chromosome that metalloproteinases are on. And I'll show you a variety of different population genetic parameters that tell us something about how genetic diversity is sorting in these populations. First, we'll look at a measure called pi, which is a measure of within population genetic diversity. And we see that in the snake venom metalloproteinase region that there's a local increase in this measure in both species. The SVMP region here is just shaded in gray. Now we also see an associated peak in a between population diversity measure called DXY, and also an associated decrease in relative population differentiation measured using FST in the same exact location. And this is a confluence of patterns that's really fairly consistent with a maintenance of ancestral polymorphism via balancing selection in this broad SVMP tandem array. And is importantly, it's not consistent with a dominant role or a dominant footprint of the influence of directional positive selection on linked variation in this sort of large region. So with those signatures pointing to balancing selection, we wanted to look for additional evidence that selection in general has been operating on the venom region. And so we turned to a series of additional statistics to clarify the roles of selection. The first was what's called the integrated haplotype score. This is a measure that allows us to detect selection for specific haplotypes or a specific haplotype in the case of directional selection. And it does this in a way that accounts for background recombination rate, which is the reason I mentioned that will become apparent in a few slides here. And the magnitude of this value gives the further it gets from 0 gives us some sense of how strong selection has been in this region. And so we see that the SVMP region, again, here shaded in gray, is really quite outstanding. With respect to the rest of the chromosome. Down here, I'm just showing you summaries of the distributions within the venom gene region compared to the chromosomal background, but also in a comparison to non-venom homologs of our venom gene family. It seemed like kind of a useful evolutionary comparison. So this high magnitude value certainly indicates the footprints of selection, but IHS doesn't really tell us what specific mechanism has been operating. And so we used an additional statistic, handful of statistics actually, but I'll just show you some results from a statistic called Tajima's D. This is designed to detect skews in the allele frequency spectrum due to different forms of selection and deviations from neutrality. What we find in the SVMP region in our species is an excess of intermediate frequency alleles leading to a high value of Tajima's D. And this is really quite consistent with balancing selection having operated on this region. So with this stack of evidence pointing towards the footprints of balancing selection, we next wondered if balanced polymorphisms in the SVMP region have persisted throughout speciation. And if so, do any of these trans-species polymorphisms translate to amino acid variation, right? Are they actually contributing to functional variation that can be useful in expanding the venom repertoire of our species? And so we simply examined alignments of major venom gene regions, looking in both the coding and non-coding regions of the arrays, looking for shared and private polymorphisms versus fixed differences. And we just tabulated these proportionally. And what we find is that in the chromosome background here on the left, and in the SVMP region specifically, we find that the overwhelming majority of polymorphisms are private. They're unique to one species or the other. This is totally what we would expect to find. But what's really neat, and what I'll turn your attention to, is that there's an enrichment of trans-species polymorphisms, shown here in the blue part of the pie chart here, in the SVMP region compared to the chromosomal background. And we see an associated depletion of fixed differences between our species in this region as well. And so there's a statistical enrichment of these trans-species polymorphism within the venom region. What's even cooler is that a small handful of these actually do contribute amino acid replacements. If we look at the polymorphisms that's being maintained, there is a change in the amino acid sequence. And what's even cooler is that if we scan across our other major venom gene tandem arrays, we see that, again, a small handful of these trans-species polymorphisms contribute to amino acid variation, expanding the potential functional diversity of venom in these species. Okay, finally, with respect to balancing selection, we were curious if specific mechanisms of balancing selection have shaped venom adaptation through time. There are a whole variety of different balancing mechanisms. It's a potentially large list, but we thought of two that were really quite consistent with the ecological context of predator-prey antagonism. The first is heterozygote advantage. That's where it's exactly what it sounds like. Heterozygous genotypes enjoy a fitness benefit relative to homozygous genotypes. And then the second is negative frequency dependent selection, in which the fitness of an allele is a function of its abundance in the population. And so typically, because it's negative frequency dependence, rare alleles will enjoy a fitness benefit because in the context of venom, they've not been tested yet by prey species. They've not been observed by the resistance mechanisms. And so they'll move towards fixation until they become the major allele, in which case the process will start all over again. And so over time, that will produce a signature of balancing selection. And so we performed a simulation study to test these alternatives and to test them against selective neutrality as well. We performed forward time simulations using SLIM, calculated a whole suite of summary statistics using commustats, and we compared these evolved sequences under these idealized scenarios based on our empirical population parameters to calculate a probability, a log odds probability of these alternative mechanisms influencing genetic variation in the venom regions. And we find this here, we're scanning along the SVMP region. We're zoomed way into the venom cluster itself. Here are the genes up here and their orientations. And anywhere that you see values above the 0 line, those are greater log odds of balancing selection. And neutrality here is below the 0 line. And so, awesome. We see that based on these simulations, we're finding that a large number of these intervals are consistent with balancing mechanisms having operated. Excuse me. Then if we distill this further, for any of these intervals that are showing evidence of balancing selection, we can test whether there's a greater probability of negative frequency dependence or heterozygote advantage. And what's really neat is that we're finding that in different intervals, you have a greater probability of either or. It seems like both mechanisms to some degree have influenced variation in this tandem array. So full stop, if you're like me, when I was running these analyses, I contended with the question of how selection can operate efficiently in these regions to begin with. So venom clusters are tightly physically packed clusters of genes, and so it stands to reason that all else being equal, selection at one locus could easily interfere with selection at another. Greater physical linkage implies greater selective interference. Well, to address this, we could think back to a study from a couple of years ago in which we investigated the recombination map of rattlesnakes, and we found evidence that recombination rate is actually fairly concentrated in genic regions, such that if you average across the whole genome, as you move up and downstream of gene regions, you see recombination decrease. And often recombination hotspots, these punctuated loci where recombination happens much more often than other regions, these are usually smack dab in promoters of genes. And so this led us to hypothesize that venom tandem arrays may function as recombination hotspots. And sure enough, looking at population scaled recombination rates in which we've actually normalized for regional variation in effective population size, we see that these are significantly higher in the SVMP region than non-venom genes and the chromosome 9 background. And just as one other way of looking at this, we looked at the decay of linkage disequilibrium as a function of physical distance between pairs of SNPs in the SVMP region and in flanking regions. And we find that LD decays much more rapidly in the venom region than these flanking locations. And so this rapid decay of LD certainly supports a role of recombination in increasing the efficiency of selection in venom gene regions, kind of addressing that sort of puzzle of how selective interference can be circumvented. So I believe I'm just about out of time here. So I'll just wrap up by saying this study, and hopefully I've shown you that we have a fair amount of evidence for a maintenance of ancestral venom polymorphism via long-term balancing selection. This is, again, this sort of A plus B selective scenario. Although I will say that our results are not inconsistent completely with directional selection having operated on specific sets of genes. PLA2B notably showed really great evidence of directional selection. And so the kind of irony is that balancing mechanisms actually maintain diversity that directional selection could later on act upon. And so it's still a little bit of a puzzle, but it seems like there's a diversity of selective mechanisms that likely operate on these tandem arrays, and that balancing mechanisms maintain an adaptive genetic diversity. Recombination plays a crucial role in shaping venom adaptation through efficient selection. And the presence of balanced amino acid polymorphisms indicates to us that snakes have expanded their sort of venom toolkit or repertoire, whatever you want to call it, via gene duplication as well as functional allelic variation. We like to say that they're bringing a missile to a knife fight. And then finally, our findings are quite consistent with central tenets of antagonistic coevolutionary theory and may help to explain how snakes are keeping pace with their prey so that they don't lose the race, as it were. So again, I just want to thank my co-authors on this study, natural history collections that loan tissues, as well as funding sources. And again, a huge thank you to Ronald and the other EUVEN organizers. Thanks so much for having me. And I'd be happy to take any questions.
Wonderful. Thank you, Drew. That is very interesting. If you could unshare your screen, we'll see if there's any questions. So people, you can either type in the chat or raise your hand or even speak up, because I can't see all the little thumbnails. So any questions or comments? I wish that I could do population genetics. You can address really cool questions with this.
Anyone can do it, Ronald.
Oh, with given enough training and stuff, right?
Yeah, it's a fun angle to approach these sorts of questions from, for sure.
Just wondering, naively, clearly, when you're talking about snake venom metalloproteases, that's one of the venom components probably mostly used, in predation interactions. Do you think that similar stories could be told for venom components, maybe in other taxa to do mostly with defensive roles, or is that too much arm waving?
That is a really great question. I don't know. I think that it's a different ecological context, and I don't know if it'd be the same. I mean, because there your constraint is, are you good at keeping something from killing you? And if you can meet the minimum requirements to do that, it might mean that. And it might depend on how many things are trying to kill you. So if you're being predated upon by birds and large mammals or what have you, I mean, it could be a different story, but that's a whole other avenue that I hadn't even considered. But because I'm less well versed in the invert world, but I understand that there are some venoms that are like explicitly defensive,
Yes.
and so that'd be a really interesting line of inquiry for sure.
And of course, so some, if you look at certain groups, mention assassin bugs where they have venoms that are both defensive and predatory. I mean, they're created by the same organism. There's a different cocktail of components. The same with some of the cone snails. So your story already shows very fascinating complexity. It will be even more complex in such systems, I suppose. I've got a typed question from Icy. It was a good talk. Thank you a lot. I wonder, what is the gene polymorphism expression level between juvenile adults and male-female snakes? So I guess is there differences between ontogenetic stages and sexes?
Shorter answer, I have no idea. We only went as far as to assess whether there was functional diversity in terms of those amino acid replacements. We've done no modeling. We've done no gene expression analyses of those different alleles. I mean, that's entirely open territory. It's not a very satisfying answer, but that's sort of what we could fit in the timeline of the study and in the context of the paper. But that'd be a really cool thing to do. And I know that other people who are much more well suited to investigate the ontogenetic shift stuff. I think there are folks that are working on that, but how that relates to the polymorphism would be a really cool thing to investigate.
Always more questions. I'll do a final question typed in by Lorenzo Senizzi. He says, how generalizable do you think your results are for these two snake species? And he's specifically thinking about Mojave rattlesnakes, where the combined neurotoxic hemotoxic phenotype is selected against rather than maintained across populations, to my limited knowledge. Amazing talk. If any ideas?
Yeah, I'll be curious. Well, I don't know. There's never enough time, but I have an idea for why the A plus B venom is selected against, and I think it has to do with genetic incompatibility. There's not enough time to explain why, but we do know that the species that are neurotoxic, that possess the specific SVMP haplotype that includes a huge deletion, there are a lot of reasons to think that there could actually be basically like disjunction or something like that could occur during meiosis. Mm-hmm. That's a little bit of pie in the sky stuff, but there is some results a couple of years ago from one of Mark's collaborators, Jason Strickland, that totally convinced me that was the case. I can't prove it, but it'd be cool to do that someday. But with respect to when that isn't the problem, we also need to consider all of the other things that need to come along with what it means to be neurotoxic versus hemotoxic. Yeah. We understand that the snakes are inheriting not just the toxins, but the things that allow them to package those components in and store them safely and all that. And that's like a whole other can of worms that is an exciting can of worms, but complex nonetheless.
Excellent. I'm not going to text you further. Thanks for your wonderful talk. Congratulations on that cool Nature paper. It's near the top of my in tray, but now I have a very good impetus to read it, because if this talk lingers a bit, I may understand more of it. So that's all good.